Okay, so this is the screencast on the second part of protein synthesis, the actual synthesization, if that's a word, of the proteins. So basically what happens is, as we talked about in the last video, your your mRNA, the messenger RNA that was transcribed from the DNA, has entered the cytoplasm and exited the nucleus. And so in the cytoplasm, it goes to one of two places, either either something called a free-floating ribosome, or a ribosome that's located on the rough endoplasmic reticulum, also abbreviated as rough ER. And the differentiation is if the mRNA is going to stay in the cell that it's been synthesized in, if it's going to stay, if the protein is meant for something going on in that cell, it's going to go to a free floating ribosome. And if it's going to be transported outside of that cell to another cell, so like some sort of cell communication, then it's going to go into the rough ER, or it's going to go to a ribosome in the rough ER. But basically what happens is the same thing. So it goes into the ribosome. There's two parts of the ribosome. There's this top part here, as you can see. This is really thick green and I do not like it. So I'm going to make it not so thick and I'm going to make it black. Anyways, as you can see, there's this huge long strand of mRNA, and there's this top part of the ribosome and the bottom part, and it kind of just goes like in between. Like there's this little, I guess it's kind of like a synaptic cleft if you're familiar with like nerve cells. It's like this little space that it goes through, and there's these tRNAs, and basically what they do is when we were taking notes on it in class, it looked like little sombreros, and he drew them like this. It was actually kind of cool. And then here would be the protein, and then they'd have three unique nitrogen bases. So see here, it's just, the same thing. There's this protein here, whatever it is, and then three nitrogen bases, and it comes, and as you can see, this A is going to pair with the U, the G with the C, and the C with the G, and it's going to fit perfectly. And basically what happens is, once it attaches here, once this becomes it becomes bonded and they match up, then this tRNA disappears and it's no longer there, and it's just the protein that's left. And then the proteins attach to each other in something called with something called a peptide bond, which is just a bond between two proteins. And it's also good to remember that a peptide bond occurs between the NH2 group and the carboxyl group, which is COOH. But I don't have any room to write that. Let's see, COOH. Whoa. Okay, close enough. So that bond happens there, and that's a peptide bond. We'll do that in a different color to make it more. There we go. Well, still not good enough. There we go. So that there is a peptide bond. And so what happens is, after a whole bunch of these are bonding together, like this bonds to this one, and these are all amino acids. This amino acid bonds to this amino acid bonds to this one, and there's a whole chain of them. It becomes a poly, which is the prefix of many, so it becomes polypeptide chain. So it's just this chain now of proteins, and it goes wherever it needs to go. So that's basically the end of how protein synthesis is synthesized, I guess. It just goes through this ribosome, and these tRNAs match up with, it has to fit perfectly. Like this, if this was a U or something, or an A, then it wouldn't be this specific protein. Like it'd be another protein completely. So it's important to remember that it has to have the exact three nitrogen bases in order for it to have that specific amino acid coded for it. And so that's pretty much it. Thanks for listening, and I hope I get an A on this website. Alright, awesome. Bye. Oh, also, biology is awesome. Okay, bye.